Hi all, I'm so happy you were able to make it for today's lesson. Today we'll be focusing on the regulation of glycolysis and gluconeogenesis, with particular attention to the role of Aldolase. We will first focus on how glycolysis and gluconeogenesis function and how these opposing pathways are regulated. Then we will shift our focus to the Aldolase and examine why the same enzyme can participate in both pathways. From there, we will focus on the different Aldolase isoforms and where they are expressed throughout the body. Towards the end of our focus, we will begin to narrow to Aldolase B and examine how its expression is regulated by the transcription factors HNF1, HNF3, CEBP, and DPB. However, before we begin, let's review our lesson objectives of what you should be able to describe, identify, and explain by the end of today's lesson. Before we get into our pathways, we're going to go over the 4 main objectives that's going to help us guide our lesson for today. First, we're going to review our major steps and regulation of glycolysis and gluconeogenesis. More importantly, we're going to discuss how these pathways are regulated. Since they essentially accomplish opposing metabolic goals, we need to understand how the cell coordinates their activity. Next, we are going to focus on Aldolase and look at the reaction it catalyzes in both pathways. Then we are going to compare our 3 major Aldolase isoforms, Aldolase A, Aldolase B, and Aldolase C. And we're going to connect their expression patterns to different tissues within the body. Finally, we're going to move from our metabolic regulation to our gene regulation. We are going to focus specifically on Aldolase B and examine how HNF1, HNF3, CEBP, and DBP influence its expression. As we also move through the slides, you're going to see, because we start with pathways, then go into Aldolase, then isoforms and gene regulation, where you're going to see these tags on each and every slide, depending on what we are discussing. So we are going to now start with the pathway that allows cells to extract energy from glucose, which is glycolysis. So on the next slide, you're going to see this pink tab. This is to help you formulate and visualize where we are at within the process. Now let's begin with the first half of glycolysis. This is known as the preparatory phase. Again, glycolysis is a pathway, so we have our little pathway sign down here. Glycolysis occurs in the cystosol, and during this first phase, the cell actually has to invest energy before it can begin capturing the energy from glucose. So we're going to start at the top of this figure here, okay? We're going to begin with 1 6-carbon molecule of glucose. And in the first reaction, hexokinase transfers a phosphate group from ATP to glucose, producing a glucose 6-phosphate. Okay, let's back up. I want you guys to understand that we are using ATP here instead of producing it. This is the first ATP investment of glycolysis. Now let's move on to step 2. In step 2, phosphohexose imerase rearranges the glucose 6-phosphate into fructose 6-phosphate. We still have a 6-carbon molecule. However, at this point, we simply rearranged its structure in preparation for the following reaction. So here in step 3, phosphofructokinase 1, or PFK1, uses a second ATP to convert fructose 6-phosphate into fructose 1,6-biphosphate. So now at this point, the cell has invested 2 ATP molecules and has not used up any ATP yet. PFK1 is still an important regulatory enzyme within glycolysis. So it's important to remember this step because we're also going to return back to it when we discuss how the pathway is actually controlled. Okay, so now let's look at our fructose 1,6-biphosphate because that's going to lead us into step 4, okay? So now we're reaching an enzyme that's going to become especially important later in the lesson, Aldolase. Aldolase cleaves the 6-carbon fructase 1,6-biphosphate into 2 carbon products, glycerol hydride 3-phosphate or GAP or DHAP. So now moving down here, here's our 2 molecules. Here's our GAP, GAP or DHAP. So what's happening to our carbon skeleton? Well, we started with 1 6-carbon glucose molecule, and now we have divided those carbons into 2 3-carbon molecules. But there's a problem. Only 1 GHP continues directly into the next phase of glycolysis. So this is a reverse arrow. So triose phosphate converts DHAP into another molecule of GHP. As a result, for every 1 glucose that is entered into glycolysis, we now have 2 molecules of GHB moving forward into the payoff phase, which we're going to see on our next slide. But this is important because from this point forward, the reactions that we're going to see occur twice for every glucose molecule, okay? So let's take it back and let's summarize where we are. We have taken 1 6-carbon glucose molecule. We have invested 2 ATP and produced 2 3-carbon intermediates. But so far, we've only spent our energy instead of gaining our energy. Okay? So here's a question I want you to think about as we move forward. If glycolysis has already used 2 ATP, how can the pathway ultimately result in a net gain of energy? Well, keep those 2 GAP molecules in mind because on the next slide, we're going to follow them through in the payoff phase and answer that question. So on the previous slide, I asked you to think about how glycolysis can produce a net gain of energy when we already invested 2 ATP molecules. Well, the answer is going to come from what happens during our payoff phase. This is the second phase in glycolysis. We already went over the first, which is preparatory, and now we are in the second, the payoff phase. So we're going to start here, which is where we ended up in at the end of the first phase. We're going to start with our GAP molecule, okay? Preparatory phase gave us 2 GAP molecules from our original 1 glucose molecule. This is important because every single interaction that we look at on this slide occurs twice for each glucose that is entered into glycolysis. So let's move our attention to step 6. So the payoff phase is going to begin with what we call oxidation of the GAB. At step 6, GAP catalyzes a reaction in which NAD is reduced into NADH. Since this reaction occurs twice, we then generate 2 NADH molecules per glucose. This allows some of the energy released during oxidation to be captured in an electron carrier instead of being lost. So it's not lost, it's now just being stored in an electron carrier. So here in step 7 is where we're going to start to answer our own question from the previous slide. At step 7, phosphoglycerate kinase transfers a phosphate group directly from 1,3-biphosphate glycerate to ADP, producing our ATP. This is called our substrate level phosphorylation because ATP is being generated through a direct transfer of a phosphate group from a metabolic intermediate to ATP. Since we have 2 3-carbon molecules moving through the pathway, 2 ATP molecules are then produced. But do not forget, we spent 2 ATP during the preparatory phase. So at this point, we've only recovered our original ATP investment. We have not reached a net ATP gain yet. The pathway is then going to continue as the 3-carbon intermediates are rearranged. So that's going to happen in step 8 and in step 9. And this is then going to eventually produce our PEP, phosphofoleric pyruvate. Our PEP is going to contain high energy phosphate group. Therefore, at step 10, pyruvate kinase transfers that phosphate to ADP, producing another ATP through the substrate level, phosphorylation, and leaving pyruvate as the final product. Because this reaction also occurs twice, another 2 ATP molecules are produced. So we have our 2 ATP molecules here in step 7, and again here in step 10. So now we're able to answer our original question. During the payoff phase, glycolysis produces 4 ATP. We invested 2 ATP during the preparatory phase. So 4 ATP minus 2 invested ATP gives us a net gain of 2 ATP per glucose. In addition, glycolysis produces 2 NADH and 2 pyruvate molecules for every glucose molecule that enters the pathway. Pyruvate then can have different metabolic fates depending on the cell and the availability of oxygen. However, for our purposes and for this lesson, we have now followed glucose through the whole complete 10-step glycolytic pathway. There is one more feature on this figure that I want you to notice. Look at pyruvate in step 10. Pyruvate kinase is one of the enzymes involved in controlling glycolytic flux. Earlier, we've also encountered hexokinase and PFK1. These regulatory points are important because a cell does not benefit from breaking down glucose at maximum speed, regardless of its energy that it needs. So here's the question to think about before we move on. If glycolysis produces usable energy for the cell, why would the cell ever want to slow down the pathway? Well, to answer that, we're going to need to look at how glycolysis responds to the cell's energy, status, and how its major regulatory enzymes are then controlled. So, on the previous slide, I asked, why would the cell ever want to slow glycolysis down if the pathway produces usable energy? Well, the answer is glycolysis needs to respond to the energy needs of the cell. If ATP is already abundant, there is little benefit to continuing to break down glucose at the same rate. However, when the cellular energy is low, increasing glycolytic activity can actually help meet that demand. Regulation is concentrated at several important steps in the pathway. Now, this figure does not have our little pathway bookmark sign, but it is the same color to show that this is a part of our pathway. So, our first regulatory port is our hexokinase here. This is at the very beginning of glycolysis, and it converts glucose to glucose 6-P. In many tissues, accumulation of glucose 6-phosphate inhibits hexokinase. This is an example of product inhibition. If glucose 6-phosphate is accumulating rather than continuing through metabolism, the cell can then reduce the amount of an additional glucose entering glycolysis through this step. Also to note, this is a chart that I would write down in your notebooks. Instead of giving another difficult model, I chose to make it into a nice, easy table for you for reference. So now moving on to the PFK1. This is the most important regulatory point to focus on. However, PFK1 catalyzes the conversion of fructose 6-phosphate into fructose 1,6-biphosphate. This is a major control point for glycolic flux. If you look at the regulators on the right, ATP inhibits PFK1. At first, that might sound strange because ATP is also a substrate in this reaction, but ATP has two roles. At lower concentrations, it's going to provide the phosphate required for the reaction. When ATP becomes abundant, it's also going to act as an alloristic signal, indicating that the cell's energy supply is already high. Citrate provides another signal of abundance metabolic fuel and also will inhibit PFK1. In contrast, increasing AMP and ADP indicate a lower cellular energy state. These signals favor PFK1 activity and therefore greater glycolic flux. PFK1 is also strongly activated by fructose 2,6-biphosphate. The most important thing for you to remember is that this regulator in particular, we're going to return to it. Because after we introduce gluconeogenesis, fructose 2,6-biphosphate will then help coordinate the activity of these two opposing pathways. So now we're going to move on to our third and final row, pyruvate kinase. This is a third major regulator enzyme. This enzyme, pyruvate kinase, is going to catalyze the final steps of glycolysis. High energies of ATP and alanine inhibit pyruvate kinase, again helping slow glycolysis when energy or biosynthetic precursors are already abundant. But in contrast, fructose 1,6-biphosphate is then going to activate pyruvate kinase. This is an example of feed-forward activation. An intermediate is produced earlier in glycolysis, which signals a later enzyme that is a substrate able to move through the pathway, helping coordinate activity between earlier and later reactions. So instead of thinking of glycolysis as a pathway that simply turns on whenever glucose is present, think of it as a pathway whose flux is continuously adjusted according to the metabolic state of the cell. So high energy signals generally slow glycolysis. Low energy signals favor greater glycolic activity. Okay, now let's change the metabolic situation. Imagine that glucose availability is low, but certain tissues still require glucose. How can the body produce glucose when it cannot simply obtain enough from it from the bloodstream or the diet? Well, let's find out in our next pathway. So here is our pathway bookmarks. We are still in our pathway territory. So previously I asked how the body is able to produce glucose when glucose availability is low. Well, the answer is gluconeogenesis. This is a pathway that synthesizes glucose from non-carbohydrate precursors and is especially important for maintaining our blood glucose when dietary glucose is limited. Now this is one image, however I did crop it so it's easier and more visually appealing for you all to see and follow. So this figure on the slide here that we are looking at as a whole, just split in half, compares glycolysis in red with gluconeogenesis in blue. But we are focused on the gluconeogenesis now, and we're going to follow the blue pathway upward. So we're going to begin at the bottom right hand of the figure and work our way up to the left side, okay? So we're going to start down here by the pyruvate. So the important starting point for gluconeogenesis is this 2-pyruvate. Rather than continuing toward energy production as it would following glycolysis, we're now going to use that carbon to work our way back toward the glucose. So we're going to start at our pyruvate. immediately we encounter a problem. The pyruvate kinase reaction we saw at the end of glycolysis is essentially irreversible under cellular conditions. So gluconeogenesis cannot simply use pyruvate kinase in reverse. Instead, pyruvate is the first converted by oxopyruvate carbolase, which requires ATP shown here. Then the oxoacetate is here. So oxoacetate is then converted to our PEP, carbokinase. This reaction uses GTP, and together these reactions provide a bypass around the irreversible pyruvate kinase step of glycolysis. So we start with our pyruvate carbolase, oxocarbonate, and then our PEP carbokinase, okay? So once we are now past this barrier, we notice something, again, important through this central portion. The blue and red pathways essentially follow the same route, but in opposite directions. That's because many of these reactions of glycolysis are reversible, and the same enzymes are able to participate within both ways. So as we continue upward, we're moving toward the 3-carbon intermediate that we saw earlier in glycolysis. Here, our GAP and our DAHP are used to form our fructose 1,6-biphosphate. Notice which enzyme is involved here. Our Aldolase. Earlier, we watched Aldolase cleave fructose 1,6-biphosphate during glycolysis. Here, the same reversible Aldolase reaction operates within the opposite direction. We're going to come back to exactly how the Aldolase functions within both pathways later. But now we're going to continue to move up and move forward. So now we're going to encounter our second irreverse glycolic step. PFK1 cannot simplify, operate backwards. Instead, glucogenesis uses fructose 1,6-biphosphate to convert fructose 1,6-biphosphate to fructose 6-phosphate. So we see this here, and it's moving up here. Remember, we're focusing on the blue side, okay? Finally, the hexokinase reaction from glycolysis also cannot be simply reversed. Glucose 6-phosphate bypasses that reaction by removing the phosphate from glucose 6-phosphate, allowing glucose to then be produced. So now we're up here at glucose. Therefore, we can now answer our own question. When glucose availability is low, gluconeogenesis allows the body to synthesize glucose rather than break it down. But I want you to know that we had to take a different route at 3 separate locations. We had to take a route here at the pyruvate. We had to take another route here at the fructose, and another route here at the glucose 6-phosphate. These bypasses show that it is an important distinction. Gluconeogenesis is not simply glycolysis running backwards. Most of the reactions are reversible and can be shared between the pathways, but the 3 essentially irreversible reactions of glycolysis require different enzymes during gluconeogenesis, and that is very important. So here's the one question I want you to think about as we continue to move forward. If glycolysis and gluconeogenesis have opposing goals, what prevents the cell from running both pathways at high rates at the same time? Okay, so we are officially out of our pathways, and we have moved on to Aldolase, as you can see up here by our little green bookmark. Okay, so on the last slide, we asked if glycolysis and gluconeogenesis have opposing goals, what prevents the cell from running both pathways at high rates at the same time? Well, the answer is going to be reciprocal regulation. Again, I could have put a very detailed large image up here with all the arrows and the words. We're not doing that. This is something that I want you to remember, and this is the easiest way to remember it is if it is broken down in layman's terms, okay? So this is something that I would 100% write down. So based off of the reciprocal regulation. So how does that happen? The cell is then coordinating these two pathways. So when conditions favor glycolysis, glucogenesis is not generally suppressed. And when conditions favor glucogenesis, glycolysis is then reduced. This is important because at the irreversible regulatory steps that we identified earlier, by regulating opposing pathways in opposite directions, the cell is then avoiding wasting energy through what is called the futile cycle. The futile cycle is where glucose would essentially be broken down and rebuilt at the same time without accomplishing useful metabolic work. But there is a very important distinction that I want to make clear. Not every reaction in these pathways need a separate enzyme. Most of the reactions that we have saw so far are reversible and are actually shared between glycolysis and gluconeogenesis. But instead of looking at another overly complicated pathway diagram, like I stated earlier, I really wanted to break this next concept down into simpler terms for you because, again, this is important and I highly recommend writing this down. Okay, so this is one of those standard shared reactions. And the enzyme that we're talking about is on Aldolase. Okay, we're focusing on our Aldolase. This arrow is just showing you that the Aldolase is going to be within that reversible arrow, okay? So this reversible arrow here is important because Aldolase can catalyze this reaction in either direction depending on which pathway is operating. So let's look at glycolysis first, okay? So during glycolysis, Aldolase catalyzes the cleavage of fructose 1,6-biphosphate into 3 carbon products, GAP or DHAP. We saw this exact reaction earlier during the preparatory phase of glycolysis. And in this direction, we're taking 6 carbon molecules and splitting it into 2 3-carbon molecules, okay? So now let's look at the reverse. Let's look at our gluconeogenesis. So during gluconeogenesis, Aldolase catalyzed the condensation of GAP and DHAP. Now we're taking those 2 3-carbon molecules and combining them as a pathway that works by producing our glucose. So this reversible arrow really just summarizes the major concept on this slide. Aldolase participates in both pathways because the reaction it catalyzes is therefore reversible. The reaction moves towards the cleavage during glycolysis and toward condensation during gluconeogenesis. But here is where Aldolase becomes even more interesting. Humans do not just have one form of this enzyme. We have different Aldolase isoforms with distinct patterns of tissue expression. So now let's think about it. If Aldolase isoforms catalyze similar reactions, why would different tissues need different forms of this enzyme? So if Aldolase isoforms catalyze similar reactions, why would different tissues need different forms of this enzyme? Well, the answer is going to begin with their tissue-specific expression. patterns. Keep in mind, we are still discussing our Aldolase. So humans have three major Aldolase isoforms: Aldolase A, Aldolase B, and Aldolase C. They are not distributed uniformly throughout the body. Okay, so this table does have a lot of information on it. Do not panic. We are not going to try to interpret every single column. Instead, I'm going to highlight just the information that is important to what we are discussing. Okay, so we're going to start here with our organ column. So we're going to look at all of our organs down in this area. Okay, this column is going to tell us which tissues are being compared within the study. Now we're going to highlight another column, and it's called the second enzyme of metabolism. Okay. This column is going to identify the Aldolase isoform associated with fructose metabolism in each of these tissues. So if we were to compare these two highlighted columns, a clear tissue-specific pattern is going to begin to emerge. Okay, so we're going to start here with the intestine. It's going to be the first one. Okay, starting with the intestine, we see Aldolase B, labeled ALDOB. Then let's move on to our liver. That's the second one in the column. Okay, we again see Aldolase B. This is particularly important because the table identifies the liver as handling approximately 50 to 70% of metabolized fructose, making this a major site of fructose metabolism. Now, let's move on to the kidney. Third one. In the kidney, we see the same pattern, Aldolase B. So already the Aldolase B is standing out across three different tissues that the source identifies as the primary sites of fructose metabolism. Let's keep moving down. We're going to move to our muscle now. But when we move to the muscle, the pattern changes, and this table identifies Aldolase A instead of our Aldolase B like it did in the intestine, liver, and kidney. Well, let's keep going. Can't stop now, right? So let's move on to our red blood cells. Our red blood cells show Aldolase A. Very interesting. And let's go to our brain, which is down here, fourth from the bottom. In the brain, we see another isoform, Aldolase C. Okay. And the table also identifies Aldolase C in adipose tissue. So rather than having one aldose isoform distributed identically throughout the body, we see a clear pattern of differential aldose expression across different tissues. Okay, so this helps answer our question from the previous slide. Different tissues, as seen here, have different metabolic roles and demands. Therefore, their aldose expression reflects their specialization. Most importantly, for the remainder of our lesson, Aldolase B is prominent in the intestine, liver, and kidney, and these are the primary sites of fructose metabolism identified in this source. We are going to focus especially on Aldolase B from this point forward because it is tissue-specific expression, is not accidental. The Aldolase B gene itself has to be regulated. Therefore, that appropriates tissues producing this isoform. But before we can get into the regulation, this is our next think about question. What is different about Aldolase B that makes it particularly important in tissues like the liver? Well, to answer that question, we're going to now focus specifically on fructose metabolism. Also, please note that we are on our isoform bookmark now. So this figure will look complicated at first, but we're not going to follow every pathway shown. I want you to focus on the route that begins with fructose. because this is where aldose B fits into it, okay? So focusing on fructose here on our far left side. We're going to start here, and in tissues that metabolize fructose, it's going to enter a pathway that is distinct from the initial steps that we saw prior in glucose that entered into glycolysis, all right? So starting with our fructose, we're now going to take a trip over our arrow, and this arrow is showing the catalyzation by ketohexokinase, also known as KHK. Then from that catalyzation, we end up at fructose 1P. So our KHK phosphorylates fructose to produce fructose 1-phosphate, aka fructose 1P. Now the next step is the one where I want you to pay very close attention to. Here is where Aldolase B becomes particularly important. Within the tissues we identified on the previous slide, Aldolase B functions in fructose metabolism by cleaving fructose 1-phosphate. The reaction produces 2 to 3 carbon molecules. It produces our GA, glycerol aldehyde, and our dihydroxyacetate phosphate, DHAP. But we're going to now connect this back into a simpler reaction that we saw back on slide 7, okay? So earlier we saw Aldolase acting on fructose 1,6-biphosphate during glycolysis and gluconeogenesis. Aldolase B is very distinctive because it also has an important role within the fructose metabolism through its activity with fructose 1P. So let's look at what happens to these products. So if we follow our aldo through our GA is then converted to our glycerol 3-phosphate, or our GA3P, okay? Now let's go to our DHAP. Our DHAP should already look familiar. It's one of the 3 carbon intermediates we encountered in glycolysis and gluconeogenesis. So fructose metabolism isn't operating completely independently from the pathways that we have already discussed. Instead, its carbon can never enter the broader network of central metabolism. So now we're at our pyruvate. Okay, so fructose to KHK, fructose 1P, aldo, GH, DH. So our pyruvate can contribute to the formation of acetylcholeic acid, which is down here. And from there, we have our TCA cycle, which contributes to the energy metabolism because carbon is therefore able to enter. But notice there's another possible direction shown in the figure. There's an arrow from acetylcholine A to fatty acetylcholine A to then triglycerides. So carbon is also able to enter the direction of fatty acetyl-CoA and triglyceride synthesis. This shows us that fructose-derived carbon can contribute to several different metabolic outcomes rather than just having one singular fate. So now let's return up here to our fructose 1P. For our lesson, this is the portion of the figure I want you to remember. Fructose is then converted with the catalyzation of KHK into fructose 1-phosphate. And then Aldolase B helps process that fructose 1-phosphate into 3 carbon products that connect with the central metabolism. This helps explain the tissue pattern that we saw on the previous slide. ALDO-B is especially relevant in major fructose metabolizing tissues because it provides an important connection between the fructose metabolism and the metabolic pathways that we have already been studying. So this is where our important shift in our lesson occurs. We have already looked at Aldolase and what it does and where it is expressed. Now we need to determine how cells control whether the ALDO-B gene is actually expressed. So let's think about our liver again. When does our liver cells know when and where to turn on our ALDO-B gene? Is it just there? Is it under certain circumstances, certain environmental stressors? Where does that transcription factor come into the picture? Well, let's try to find out. So, how does a liver cell know when and where to turn on our ALDO-B gene? Well, to answer that, we need to move upstream from the enzyme itself and look at the promoter gene of the ALDO-B gene, which takes us into our gene regulation bookmark, our last bookmark of our lesson. So earlier work of Raymond Jean and his colleagues helped categorize the regulatory regions of the Aldolase promoter and identified binding sites for liver-enriched transcription factors. Therefore, building on that network, Gregory and his colleagues then examined how these regulatory sites and their associated transcription factors actually influence ALDO-B promoter activity. So, up here we have our DNA sequence. This figure is derived from Gregory and his colleagues' research. So therefore, this is going to help allow us to connect all of those dots from Raymond Jean and Gregory research. So Gregory and his colleagues identified the binding regions with their functional effects on the promoter activity. There is a lot happening in this figure. We do not need to memorize the DNA sequence. What I want you guys to focus on is who binds where and what happens to promoter activity when these regulatory regions are changed, okay? So our researchers identified that several regulatory elements within this relatively short region of the promoter extends from approximately -190 to -103. So let's start here. This region can interact with DBP and CEBP. These are two of the transcription factors that we're going to examine as regulators of ALDO-B expression. Now look at this region here. We have HNF1 and HNF3, which bind to overlapping sites within the promoter. That is important because sharing an overlapping binding region creates the potential for these transcription factors to influence another's ability and to regulate that transcription. Gregory specifically investigated this relationship. So the ALDO-B expression is not controlled by one transcription factor acting as a simple on-off switch. There's multiple regulatory elements and multiple transcription factors contributing to the promoter activity. Now, let's look at the experimental portion of this figure. So we're going to now be focusing on the bar graph down here. The researchers tested different versions of the ALDO-B gene promoter using the reporter system and measured the CAT activity shown here on the left-hand side of our graph on the y-axis. For the purposes, we're going to use the height of these bars as an indication of relative promoter activity. So a higher bar is going to represent greater activity from that particular promoter. So this -190 construct contains the promoter region we've just been looking at and provides an important reference point. As different portions of the promoter are altered or removed, Notice the promoter activity then changes substantially. That tells us that these DNA regions aren't simply structural, they have functional regulatory roles. I especially want you to look at when we approach this region. Now we're going to look at the bar that is negative 126 and then to negative 113. So when the promoter was shortened to negative 113, removing region containing HNF1 and HNF3 binding site, promoter activity became essentially inactive. The researchers found that the mutation of HNF binding site decreased promoter activity approximately fourfold, which is a lot compared to the corresponding inact promoter construct. Now let's move back up to the HNF1. So this gives us an experimental evidence that this regulatory region is particularly important for the ALDO-B promoter. But here's where the regulation becomes more interesting. HNF1 and HNF3 can recognize our overlapping sites, but that doesn't mean that they have identical effects on the transcription. In fact, it was found that these transcription factors can have very different effects on the ALDO-B promoter activity. So now let's think about this. It's if HNF1 and HNF3 bind to overlapping sites on the ALDO promoter, do they cooperate or are they competing to control its own expression? Well, let's go investigate that next with the experimental results. So let's find out when HNF1 and HNF3 interact with those overlapping regulatory sites. This experiment is going to help us answer that question from these results. So this graph is very dense. So instead of looking at everything at once, we're going to just move through the three experimental groups from left to right. So we're going to start by looking at our labels. We have HNF1, HNF3, and DBP along the bottom left of the figure, and each one has different numbers. So first, these labels are going to tell us which transcription factor were introduced in each experimental condition. These numbers represent increasing amounts of HNF1 or HNF3 expression vector, while the DBP was held at the indicated amount within the experiment where it was included. Just like in the last experiment, our CAT activity is going to be our measure of promoter activity over here on the y-axis. So as these bars get taller, that's going to tell us that the ALDO promoter is being activated more strongly. So let's start with our first group on the left. So here, increasing amounts of HNF1 are tested without DBP. Notice that the CAT activity increases more as HNF1 is introduced. This tells us HNF1 is a very strong activator of the aldose-B promoter. Okay, well now let's compare that with our HNF3. Hard to see, but there are some bars here. They are very, very, very tiny, but they are here. Even as the amount of HNF3 increases, we don't see that same increase in the promoter activity. HNF3 alone did not significantly stimulate basal promoter activity within these experiments. So even though HNF1 and HNF3 can recognize overlapping regulatory sequences, they definitely do not have the same effect on the ALDO-B transcription, which is an important key takeaway based off of these results. Okay, so now let's go into the middle of our graph here where DBP is marked off. And these are not numbers, these are plus signs for anyone in the back who cannot see. So here DBP is present and increasing amounts of HNF1 are then added. We see here 0, 1, 5, 2.5, 7.5. Oh, 0.1, 0.5, 2.5, and 7.5. So we're going to look at how dramatic this promoter activity rises, right? DBP can activate the promoter, but when HNF1 is added with the DBP, the response is very much stronger. This shows us that HNF1 and DBP can work together to enhance that aldolase B promoter activity. Now, let's take a look at what happens when HNF is introduced instead. So now we're going to look at this portion of the graph where we have our HNF. We have the same numbers that HNF1 had to HNF3, and we're adding the DBP. Okay, so the DBP is still present during this experimental group here. So instead of increasing the promoter activity, we're now increasing the HNF3, and it's reducing the DBP dependent activation. So this is important because it shows us that HNF3 isn't simply failing to activate the promoter on its own, it's actually antagonizing the activation produced by another transcription factor, in this case, our DBP. Okay, now let's focus on this far right group here. This is going to directly address our questions about our HNF1 and HNF3. Here, HNF3 and HNF1 are being examined together. Remember, these two factors can interact with overlapping sites on the aldose B promoter. These results show and support a competitive relationship. HNF3 can counteract HNF1 mediated activation while increasing HNF1, progressively overcoming that inhibitory effect. Greger and his colleagues interpreted these findings as evidence that HNF1 and HNF3 compete for their overlapping bindose sites on the aldose B promoter. So the majority of the takeaway here is knowing that the transcription factor can bind a promoter, but it's not enough. We need to know what that factor does once it binds and how it interacts with other transcription factors. So for our aldose B, HNF1 promotes transcription, where HNF3 can oppose that activation. Their competition provides another level of control over how strongly the aldose B gene is expressed. Now here's the question I want you to think about. Do all transcription factors regulate aldose B at the same time and way in every type of liver cell? Our next experiment is going to show why the cellular environment itself matters. So our question from earlier is, do all transcription factors regulate aldose B the same way in every liver cell? This experiment from Gregory is going to show us that they do not. So here, Gregory and his researchers compared the effects of CBP and DBP in two different cellular environments. The goal was to determine whether these transcription factors have the same effect on aldose B promoter activity, regardless of the cells being studied. So our takeaway here is going to be the differentiation. Okay, so we're going to focus on our Hep G2 cells on the right-hand side currently. These are liver-derived cells that the researchers use to examine transcriptional regulation of the aldose B promoter. Okay, so our CBP and our DBP are located here on our left-hand side. So our CBP line is here, our DBP line is here. So we're going to focus on these results mainly. Okay. So in our Hep G2 cells, CBP had a little effect on the aldose B promoter activity, while the DBP was able to activate the promoter much more effectively. Okay. So in this cellular environment, the DBP was more effective transcriptional activator of the two. So now let's move across the figure. So we're going to look at the hepatocytes over here on the culture. So now we're looking at our primary hepatocytes, and this gives us a different cellular contact. Remember, these are the cells located within the liver. So when researchers tested the same transcription factors here, the pattern ended up changing, which is very interesting. Okay. So in primary hepatocytes, CEBP was able to activate the Aldolase B promoter. DBP, however, was inefficient at activating the promoter in these primary hepatocytes. So let's compare the two slides directly. So we can call this slide 1, we can call this slide 2, okay? So let's start with the DBP. So if we're moving our DBP in our Hep G2 cells, it's much weaker in our primary hepatocytes. Well, now let's do the opposite comparison with our CEBP, okay? So with the opposite comparison, we also have the opposite pattern. It's capable of activating in our hepatocytes, but very little in our Hep G2 sites. This is what we call and what we mean by cell-specific regulation. The DNA sequence of ALDB promoter does not have to change for its transcriptional response to change. The cellular environment influences how effective particular transcription factors regulate the promoter. So when we combine this with what we have already seen and learned with HNF1, HNF3, and ALDO-B regulation, this then becomes more dynamic. HNF1 can promote transcription, HNF3 can antagonize activation, DBP and CEBP can contribute differently depending on the cellular context. So instead of thinking of Aldolase B expression as one transcription factor, turning the gene on and off, think about it as a combined outcome of multiple transcription factors that interact regulatory DNA in particular with that cellular environment. So how can we put HNF1, HNF3, DBP, and CEBP together in one molecule of ALDO-B regulation? It's a lot, but let's try it. Let's bring all of them together. This I would recommend writing down as it has all of it together, putting it together. Now we can put all 4 transcription factors together and see how they contribute to the regulation of the ALDO-B expression, which is our overall goal, right? It's bringing all of those lesson objectives to our final slide all in one look. Rather than looking at another experimental figure, I have simplified these findings into this type of model so you are able to focus on the overall regulatory relationship and be able to flip back to it when you need it. So at the center, we have the ALDO-B promoter. The activity of this promoter is going to help determine whether the ALDO-B gene is then transcribed. Okay, this is why it is all here in one box and in the signal. Because once it is transcribed, it then can bring us to our Aldolase B. So when the promoter is active, Aldolase B transcription occurs, resulting in the product of Aldolase B. So what we have learned through today is that the promoter activity is not controlled by one transcription factor, but instead it's controlled by several transcription factors interacting with regulatory regions of the promoter, and they do not all have the same effect. So we're going to start here in the top left. We're going to have our HNF1, which functions as a very strong transcriptional activator of the Aldolase B promoter. Okay. So when HNF1 occupies its regulatory site, it can strongly promote ALDO-B transcription. However, HNF1 does not act without opposition. Here we have our HNF3. Remember, HNF1, HNF3 recognize those overlapping regulatory sites. So rather than producing the strong activation we saw with HNF1 and HNF3. HNF3 can antagonize HNF1 mediated activation. The experimental findings supported the competition between these factors as one mechanism for controlling promoter activity, which is why we have the stop arrow, not the actual arrow that HNF1. So even at the same regulatory region, the transcription factor occupies that site is able to change the transcription outcome. Okay. So now let's move on to our DBP, which can also promote our ALDO-B transcription, but the effectiveness depends on the cellular context within the experiments that we have seen. It can give a very strong activation in the HepG2 cells, but not the hepatocyte cells. So DBP is going to provide another route which Aldolase B promoter activity can be increased. There we go. There's my laser. And last but not least, we have our CEBP. This contributes to our activation. But again, we saw a different cellular pattern. CEBP showed a stronger activation in the primary hepatocytes and a lesser in the HepG2 sites. So this will depend on the cellular environment, just like the DBP. Now let's look at the diagram as a whole. This is the major concept I want you to take away from this section and this lesson. ALDO-B expression is the result of a coordinated transcriptional regulation rather than a single molecular switch. HNF1 can strongly activate transcription. HNF3 can antagonize that activation. DBP and CEBP can contribute to activation under different cellular contexts, but together all of their interactions explain how expression of the Aldolase B can be regulated in a tissue and cell-specific manner. And this is what brings our lesson together. Aldolase B is not another enzyme in metabolism. Its expression has to be coordinated with the metabolic functions of the tissues in which it is needed. We began with glycolysis and glucogenesis, two opposing pathways which must carefully be regulated. Then we saw that Aldolase participates in a reversible reaction shared by both pathways. From there, we examined the different Aldolase isoforms and their tissue-specific expressions, including the importance of Aldolase B and fructose metabolizing tissues. And now we have reached another level of regulation, control of the Aldolase B gene itself. HNF1, HNF3, DEP, and CEBP demonstrate how transcriptional factors interactions can connect gene expression with tissue-specific metabolic function. So for our final question, I want you to think about the lesson as a whole. Why might regulating the expression of a metabolic enzyme be just as important as regulating the activity of the enzyme after it has been produced? Well, that distinction between regulating enzyme activity and regulating enzyme expression is an important part of understanding how cells maintain our metabolic control. Thank you for being here with me today. I hope you had a good time, learned a lot, took some notes. Here are the references from our experiments that we have went over. And I hope you have a great rest of your day, and God bless.